Hello and welcome to this episode 1 of the Webcast Biotech Connect. Today we'll have the 3-minute thesis from Aretha Chan. Then we'll receive Pierre-Alexandre Rochette from New England Biolab to talk about the thesis and then interact with him about his company and the product they have. Finally, we'll get Alex to confess about some personal things, and we'll finish with the question in the hat. Stay with us, and this will be after this. Welcome back. Now we will start by listening to our first 3-minute thesis from master student Aretha Chan from McGill University, Montreal. Here we go.
Hello, my name is Aretha Chan and this is my 3-minute thesis presentation. Leishmaniasis is a parasitic disease caused by the protozoan parasite Leishmania. It was classified as a neglected tropical disease, but in reality, it is a disease of neglected people. Each year, there are 1 million new cases worldwide, and there are 1 billion people at risk. When humans are infected, some people are able to spontaneously heal, while others have chronic disease. This is a result of how the immune response interacts with the parasite. The Leishmania has an important virulence factor known as GP63, and we know that it has a variety of effects on macrophages, or the host human cell that it infects. It is able to prevent the macrophage from killing the parasite, therefore allowing the Leishmania to survive and replicate within the cell. The question I aim to answer with my thesis is how does the GP63 modulate the early innate immune response? We know that when the GP63 gene is knocked out of the parasite, it causes a delayed and less aggressive infection when injected into a mouse footpad. So, we injected mice intraperitoneally with the GP63 knockout Leishmania to compare the differences with the wild-type strain. We were able to look at the types of cells recruited, the cytokine and chemokine profile of the samples, the protein secreted in host exosomes, as well as the percent of cells infected over time. Surprisingly, we found that the immune response in terms of the number of cells recruited and the types of cytokines produced didn't vary much, whether or not the Leishmania expressed GP63. Neither did the contents of the exosomes vary. It seems that the difference in infection was not due to the cellular reaction. However, we did find that the mice infected with Leishmania knockout parasites, there was significantly less cells infected and less parasites found within the cells. Therefore, we believe that the cells are less able to eliminate the parasite due to GP63. Lastly, this project is important because current drugs for leishmaniasis are still toxic, expensive, and not readily available to people in affected populations. The further understanding of the pathology will allow us to develop innovating strategies to combat and prevent the disease. Thank you for listening.
Okay, welcome back everybody to the webcast. I'm with my first guest of the year in English, Pierre-Alexandre Rochette. Hello.
Hey, Guillaume.
Since we know each other for a long time, why not? Can I call you Alex?
Go ahead, man.
Perfect, Alex. Thank you so much. So basically, we will start by first telling you a couple of question and answers that we exchanged with the student, Aretha, about her thesis in 3 minutes. First thing that I've asked her was, let me look at that. Okay. What is known about the role of GP63 in the Leishmania infection and why this is considered as an important virulence factor? I was curious about that because in 3 minutes, you cannot explain much about this. So she answered to me that GP63, it's a zinc metalloproteinase that has the ability to degrade protein in the extracellular matrix to enter the cells. Basically, this allows the parasite to survive and cause pathology. Great. Second question I was wondering after listening to our thesis was, is the GP3 is really important by himself for survival in general of the parasite. So is there any issues cultivating the GP63 knockout parasites? And she answers that when all GP63 genes are knocked out, they are able to grow normally in culture at the same rate of the wild type Leishmania, and they are still able to infect cells. So that will, good things to know, to be able to analyze our results. Do you have any thought yourself, Alexandre?
I mean, it's really interesting. Most of the Leishmania labs do study GP63. It is very important. I'd love to see data that looks at not parasitic clearance in a cell, but transmission between cells. Like, that would be cool. But beyond that, like in primary cells, once infected in animals, if it's infected, what does the whole proteome looks like? But not just like proteome, but the modifications. Are they phosphorylated? Are they acetylated? It's kind of neat. I'm looking forward to see where that goes.
Yeah, sure. It's really interesting. Personally, I was also working on host pathogen interreactions, and there's a very much to do in this area. This is really important because there's still a lot of people stuck with different pathogen, including viruses, as we all aware now.
Yeah. Oh, not just that, but more and more we're finally going into single cell sequencing, and it would be really interesting to look at the evolution of transcriptomics within an infected mice or individual, but to be able to isolate the transcriptome between each individual infected cells instead of having just like a consensus transcriptomic of Leishmania infection. Like that, it's finally feasible to do it financially.
Yeah, it's true that a lot of things have evolved, and even master projects now can use tools that were just a dream for just a couple of generations ago.
It's so true. I made a post recently online where my whole first summer, before even my master's and PhD, I just wanted to clone like 3-4 fragments together and be sure that I could go through like 13 GCs in a row. And I had so many failures and everything. And to this day now, I could do the experiment within 2.5 days maximum. And so when I talk to people right now about thinking, conceptualizing projects for students, don't put yourself in your shoes 10 years ago. Look at what we can do now so much faster. Oh my God.
It's true. And other than that, what have you thought about, technical presentation of this is Aretha?
It's nice. I like how simple it can be in one slide. As a 3-minute presentation, it's great. The scientist in me wishes he could have seen the data for all the cytokines and exosomes, but I mean, clearly it's not the space for that. So the fact that it can leave me wanting a bit more, I think it's a success.
Yeah, sure. And I can add that she used exactly 3 minutes, so that was a perfect for the time. Thank you so much. So good luck to Aretha. Sorry about that. I also always have to check your name, Aretha Chan. So basically, one way to help you win this competition is to have the most views as possible on this video. So you can share with your friends, and good luck with you. Good luck to you, I mean. Okay, so now, Alex, it's now time to go to our next section of the webcast, the biotech questions. Alex, you're working for NEB now. What is your exact title, and in fact, what does it mean as your role in the company?
All right, so just for everyone, NEB being New England Biolabs. My exact title is Territory Manager. And I think that we can all agree by now that in the field of us representing a company, the title doesn't really mean anything.
That's
It
why
basically
hence my
just
questions.
means whatever is happening in my defined territory, I manage it. So that can be any one of my friends and scientists who have like questions about, can I use this enzyme for this application? There's an issue with your website. I dropped my tube. Can you send me a replacement? There's every single thing I'm in charge of, just because we're such a small company that we don't have the luxury of having different levels of, let's say, management, where I can deal with the main accounts, where generalists can deal with basic consumables. I'm really just in charge of every single request. And I guess the best way to see it is that, especially because we're a small company, my company's headquarters based in Boston needs me to be their eyes and ears on the field. What are the new technologies? What are people saying? What are they using our products for? But in return, the clients, they need me to be the face of the company because they're the only person that they see, other than, let's say, NEB at conferences, during presentations, or at booth. So yeah, whatever you think you might need me for as a scientist, I'm the person for that. And whatever the company needs, I have to do it.
Okay, and let's say I'm pretty sure that most of our viewers know New England Biolabs, but just in case, what your company specialized in, what kind of product or services are you offering?
Okay, so very short history. New England Biolabs started around like 74, and it was one scientist in Harvard University called Don Combe. And that was back in the day where molecular biology was in its infancy. Like, we barely discovered restriction enzymes and polymerases. And basically, to have access to the tools to do any type of molecular experiments, you needed to have the contact. Call a lab. Can I get your enzyme that you've purified? And that was before, when we have to natively purify each and every enzyme in existence. And what our founder decided to do was to ask Harvard, can we commercialize it and sell it to people? And Harvard said no, and so he quit to fund New England Biolabs. And until this day, the mentality of NAB has always been, let's do research, let's innovate, let's publish articles, let's keep doing scientific research like we used to do in Harvard. But because we're now privately funded, we're allowed to sell what we discover and help the people. And that's why you can still find one of our oldest catalogs with all of our enzymes in it, in all of the offices of the old PIs and the new ones.
Around what year was it funded?
74.
Oh.
Yeah, it's and our founder still goes every day to work. Let's say like from 10 to 2, but yeah, he's 93 by now. Still has the energy and the drive to do some science.
And still
so basically, like, that was like back then. What we do now, every new technology, whether we're talking about sequencing, proteomics, there's an enzyme somewhere. And so we're much further than the basic cloning. We're not just restriction enzymes. I mean, we've been celebrating our 10th year of next generation sequencing on Illumina and older platforms. We're the primary collaborators when it comes to academia, but what a lot of people don't know is that every single company that will create, let's say, new machines, new techniques, new diagnostics, they'll need enzymes. And we're also the #1 trusted collaborators when it comes to offering the right enzymes for them. So no matter how you think you could use an enzyme, we're going to be in there. So it's not just cloning anymore.
Great, great, great. Sounds good. So now we know more about your company, your job. Now that will be time to go to the more personal question in our next section of the webcast, the We Connect questions. Okay, now the We Connect questions. This is another section, another part of our webcast where I go a little bit more personal to show the human, behind the territory manager. So I have a couple of questions for you, sir. First one, you went early in science, I imagine. My question is, why have you chosen to go in that to go in science finally?
Okay, why did I choose just to do research in science?
Hmm.
The one class that I really enjoyed in high school was biology. And from that moment on, it was not even a choice. It was just wherever I felt more comfortable. And so from high school, I went CJEP in natural sciences. From there, I went in physiology in McGill because I was really intrigued by how the body used to work and the pathologies. And so the physiology program did cater to that. But then when I did my master's PhD, what interested me the most were always not basic things of, not the basic concept of how the human body works, but how it can go wrong. And so that's what really got me interested in viruses and infections, which is why I did my whole PhD path in virology. And still interests me deeply to this day.
Still no regrets?
Oh, no regrets. It's especially after 5 years of leaving the lab, I can only have nostalgia goggles, and I'm very happy where I'm at right now. None of that would have been possible without doing my master's and PhD where I did.
A lot of us, I'm including myself into the, people still working around the science, but out of the lab. There's a lot of days where we miss the labs.
We miss a lab, but luckily for me, I talk to a lot of scientists, so I get to go in the labs and see their despair. The best place for me to be is when I can give them solutions, but then they have to do the work. I have slept too many nights in the lab in my past. I'm too old for that now. So I'm at the best case scenario for that.
Since you said it yourself, you're too old. Have you noticed any evolution in the lab since you left? I know that I noticed myself that a lot of labs are, tend to organize themselves differently than they used to. For example, build big, shared labs. Have you seen a lot of evolution in the labs since you're visiting them a lot?
Yeah, so I would say that, and you really hit the right nail there. The way that we build labs right now are a lot more based on communal spaces where the number of benches that people are given can be dependent on the funds you get. So you do save a bit of money when you when you build new buildings, but it does take away from the intimacy of what I'm used to know, and also having the lab's dedicated space for radioactivity or for not cyber but ethidium bromide. And I do tend to trust a lot less new researchers or people I don't know. And so having to learn how to share a lot more space and share contaminations, I guess that can be tricky. But the other thing that I've noticed right now that is a lot more important is presence on social media for the PIs and the researchers. Just because you need to have a network of contacts to be able to ask for funds, to be able to be thought of when you write grants, and you need the reviewers to know who you are personally, because there's a lot of politics in science and you cannot stay in your bubble. You need to be out there in all the platforms as possible. And that includes, Twitter, Instagram, Facebook, wherever you can be.
Good. One last question, if you want, and that kind of category. Do you have any specific anecdote that could be a lab anecdote, something that you remember from your lab years, or could be also a work anecdote, from your territory manager years?
Oh, one of my favorite memories from grad school was when we went to a symposium in Hong Kong. That was back in 2011. So it was me and my PI, and my colleague was the PhD student ahead of me. Amazing experience for a week in Hong Kong. But the day before presenting, my mouth started hurting like crazy. And I was in a foreign country. I couldn't really speak the language. I was freaking out because I thought I had maybe like a huge mouth infection, some teeth to be taken out, had to go to a dentist. I couldn't really open my mouth while presenting my research. Luckily enough, the day after, I found a British dentist who told me that basically just my wisdom tooth were coming out. And,
oh yeah.
Oh yeah, that was the right time for that. Just not just one, like all 4 of them came out at the same time. Oh, that was painful.
Okay, perfect. Thank you so much, Alex. Now this will be the time for our last section of the show. The question from the hat. Okay, the first question, I will virtually pick a question from my series of questions in the app. First one for you, Alex. Um, do you have a specific TV show or a recent movie that you saw that you would recommend to us?
So it is a very campy and geeky TV show. It's not serious, but it's the most fun I can have every single week. Did you ever watch The Legends of Tomorrow?
Yes, I know that show.
It's take a superhero show, take a universe of superhero shows, take the B characters from everyone, put them together, and let them know that they're the B team,
Yeah,
and
and they
send them
know.
through time and space.
Yeah,
It
they are
is,
aware of it, but kind of even proud of it.
oh, it's so fun. You don't know where it's going to go every week, but it's so fun. Yeah, so
That
that's my
show, if
recommendation. In
I'm not
these days
mistaken,
of,
is
sorry?
it's kind of related or a spin-off of the Arrow and the Flash show.
It's part of the DC universe of comic book TV shows. And if we want to put a timestamp on that video, I'm in isolation because of COVID. And so I really need some entertainment, and that is the show for that.
Ah, of course, of course. I know that a lot of shows are being revisited, and personally, I'm watching some old movies, 80s movies with my kids. So they are 10 and 12, so they never heard about Rocky, but now they know all of it. Okay, we have time for one last question. Let me see. Let me see. Okay, Alex. PC, Android, or Mac, iPhone, and why?
Uh, Personally, I build my own PC that's based on Windows, but the work gives me a MacBook as well as the tablet and the iPhone. It works. Look, the iPhone and the iOS just works together, but I'm a PC at heart.
Yes.
So I have both. You know what?
You're kind of bilingual.
Why not both?
Okay, good answer. Thank you so much. So that will be all for our show today. Thank you so much, Dr. Rochette, for your help. Because this is still early stage of the webcast, so thank you very much for breaking the ice with me.
Thank you, Dr. Gayette. Don't hesitate to come to me again, please.
Okay, perfect. Thank you so much.
Entertainment here.
See you next time. Bye-bye, everybody.